In your mind, imagine a cell, the basic building block of all living things. How do you know what a cell looks like? Maybe you've seen pictures of cells in a biology textbook. Maybe you remember looking at cells with a light microscope in high school biology class. Where do our ideas about what cells look like, our ideas about what cells are, come from? In 1888, researchers at the Marine Biological Laboratory in Woods Hole, Massachusetts, started exploring fundamental questions about cells. They went out to local beaches, marshes, and the sea, where they collected toadfish, Nereis worms, sea urchins, and a lot of other organisms. They observed cells from these living things using microscopes. They did experiments and recorded what they learned. MBL researchers, driven by their unique questions and technologies, helped shape our modern ideas about what cells are. Our ideas about cells are shaped by microscopes and images. Microscopes help us see cells that are too small to see with just our eyes. Images, such as drawings, graphs, and photographs, are just as important, since scientists use them to record and share their ideas about cells. You might think scientists peer into their microscopes, look at some cells, and make an image that reflects exactly what they see, but it's not so simple as that. Scientists make choices at every step. Scientists must choose an organism to study, then they must choose how to prepare a sample to look at. They can use chemicals to highlight certain features of cells with different kinds of microscopes. Then they adjust microscope settings to magnify and resolve cells to a desired level, and different levels show different things. Finally, scientists design images to record and communicate what they learned. Before scientists ever choose an organism to study, they have specific questions they want to answer. Scientists' questions influence what they are looking for, the methods they use to find those things, and how they interpret what they see. Their interpretation shapes new ideas and questions, which go on to shape new experiments and images. Follow me on this historical journey on seeing cells in life. The earliest scientists relied on their eyes to learn about cells. In the 17th century, early scientists called natural philosophers began to record and share their visual observations of the natural world, of plants and animals and shells and rocks, using pictures. Natural philosophers considered microscopes valuable tools for enhancing their powers of sight. They used early microscopes to answer questions about the world at small scales. They investigated how small animals, like frogs and insects, reproduced. They looked for fundamental units of organization in physical matter, things they called atoms and globules. Letters and images became important tools for natural philosophers to record and communicate observations. Images were especially important for communicating natural philosophers' visions of microscopic things, because you had to look under a microscope yourself to verify another person's claims. Scientists have long relied on their eyes to learn about the natural world and to learn about cells. Microscope technology constrained what past scientists could see. Microscope technology constrains what modern scientists can see. 17th century natural philosophers used relatively simple microscopes made of 1 or 2 lenses of glass to view natural objects. These microscopes were impressive for the time, but people who made lenses and studied optics knew that microscopes could be different. 18th century researchers built microscopes with increased magnification, or how big samples appear. They worked to sharpen resolution, or how crisp samples appear to the eye. They found fixes for aberrations, or visual warping of the sample caused by lenses. As microscope technology changed, so did the visible features of microscopic objects. Changes to microscopes were important for the formation of cell theory during the 19th century. With their new microscopes, researchers saw uniform structures in the tissues of plants and animals alike. This is a microscope that MBL researcher and Nobel Prize winner Thomas Hunt Morgan used. We heard that what scientists see is constrained by microscope technology. There is another constraint. What scientists see with microscopes is guided by their ideas about the world, their concepts. We learned how in the 19th century, researchers like Schleiden, Schwann, Remak, and Virchow established cells as a basic unit of which all living things were composed, and by which all living things arose. But not all researchers had the same idea of what counted as life. Biologists believed that life existed as its own special, separate physical matter. They also believed that life was marked by motion. Their specific concept of life guided their microscopic observations. When these researchers saw a moving gelatinous substance in living cells, they called it protoplasm, and it became a top contender for the stuff of life. Why? When observed with light microscopes, protoplasm looked like it was constantly moving. Scientists interpreted that motion as directing the behavior of cells. The idea that life was its own substance and that it was marked by motion shaped scientists' interpretations of then visible cells. As microscope technology and ideas about cells changed, research about cells specialized. Some scientists formed a new discipline called cytology, focused on studying specific types of cells. Cytology relied on improved microscopes, as well as the invention of new methods for preparing cells for viewing. Early on at MBL, Edmund Beecher Wilson used microscopes to study cells during early stages of development. In his textbook called The Cell, Wilson provided detailed drawings of cells. These new preparation methods made it possible for researchers to see more clearly differences in the shapes and parts of cells from different organisms and tissues. Scientists interpreted these differences as indicating different functions that specialized cells played in organs and organisms. Remember how images became important tools for natural philosophers to share what they saw with other natural philosophers? Cytologists also depended on images. They used images to capture and define the differences between cell types as they appeared in the microscope. What they saw guided their image making, and their images influenced and reinforced their ability to see unique cell features with microscopes. During the 19th century, cell theory became more popular among scientists. Cytologists observed that cells were specialized in structure and function within individual organisms of many types. Some researchers sought to unify these observations. They asked, how do all the different types of cells arise from a single cell type? One cell become many integrated cells in a single organism. To answer these fundamental questions, scientists at the MBL went to their microscopes. They observed live cell division for many different organisms. They created many thousands of detailed illustrations to record the continuous process of division. Through that intensive image-making process, they identified patterns across organisms, defined set stages, and communicated those patterns and stages to others. In this case, images help scientists find patterns that would be near impossible to identify with real-time observations. Can you imagine trying to watch the live cell division of a starfish, a cuttlefish, an eel, and a sea urchin at the same time through 4 separate microscopes? And not only watch, but notice patterns and changes? This impossible task was made possible through the comparison of images. Most of the scientists we have heard about so far learned about cells by observing cell samples directly with microscopes. These scientists needed to collect organisms, take tissue samples, prepare the cells for viewing with various tools and chemicals, and do the looking. Before they could do any of that work, however, scientists learned about cells by reading about work performed by researchers before them. That previous work was passed down as words, numbers, and images in scientific publications like journal articles and textbooks. Before scientists ever observed a cell on their own, they were taught how to see cells by others. In this way, new observations of cells are partially shaped by the words, numbers, and images compiled by earlier scientists. This is a good thing because it means that we don't have to start over with every new generation. But it also means that scientists have preconceived ideas about how to observe cells, what to look for, and how to interpret what they see. These ideas are, in some ways, almost entirely divorced from the process of direct observation. There are multiple ways to learn new things about cells. Some researchers learned about cells through description, like when Anton van Leeuwenhoek wrote about and drew microscopic organisms, or when Charles Otis Whitman drew thousands of pictures to capture cell division. Other researchers learned about cells through systematic organization, like when cytologists classified cell types by patterns in structure and function. Up until the 20th century, researchers mostly learned about cells through description and classification. Some researchers eventually asked questions about cells that they did not feel could be answered through description or classification. These researchers learned about cells through experimentation, by manipulation. Manipulation could invoke putting cells in extreme environments, such as super hot or super salty water, and seeing what happens to the cell. Manipulation could also involve disturbing parts of cells and watching how they react. Scientists use manipulation to come up with different answers to questions about what cells are doing and why. What scientists learn through manipulation is not inherently better or more accurate than what they learn through description or classification. Rather, what they can learn is different. As experimental methods continued to develop, so did microscope technology. In the mid-20th century, researchers started to use electron microscopes to learn about smaller and smaller structures in cells. Electron microscopes, with impressive powers of magnification, produce very different pictures of cells compared to pictures made with earlier light microscopes. But cell biologists using this powerful tool faced a dilemma. How could they verify the existence of newly visible structures? If we had a detailed photograph of a jellyfish, we might be able to judge the resemblance of the photograph to the real-life jellyfish by, well, looking at a real-life jellyfish. But we cannot do the same with something as small as a cell. We can only see cells and their tiny features with microscopes in the images made with their aid. Scientists cannot verify the resemblance of cells to the real thing by looking directly at the real thing, so what can they do? Verifying the cell structures that appear in images takes time. Scientists consult past literature and work with one another in real time. They do experiments by comparing images of the same samples made with different types of microscopes and with different preparation methods. It is a process of trial and error. We have heard how prior ideas shape what scientists see, and what scientists see helps shape their new ideas. Depending on how different cells are prepared for different microscopes, scientists see different things, and therefore learn different things. New types of microscopy, like interference and phase contrast, enabled researchers to view the organelles of live cells. Dr. Scott Brady, a summer researcher at MBL for more than 40 years, studies axonal transport. Axonal transport is the process by which some organelles move from the neuronal cell body to the synapse and back. Brady uses video-enhanced differential contrast microscopy, a method developed here at the MBL, to follow organelle movement in real time. Scott uses isolated axoplasm from the giant axon of adult Thoriteuthis, the common squid in Cape Cod waters. These studies have led to the discovery of new molecular motors and helps us understand neurodegenerative diseases like Alzheimer's disease. The images of cells made with these new types of microscopy not only look different from those made with electron microscopy, but they also help scientists to learn things about organelles beyond their detailed structure. We have heard several times how images are important tools for recording and communicating ideas about cells. Before video technology, all images were static or still. Even though many researchers watched live cells move under the microscope, they could not easily communicate that movement through static images like drawings and photographs. Some researchers' ideas about cells came to mirror the static image. Many scientists thought of the structure of cells as static, still, and rigid. Video microscopy applied to cell research changed not only what people saw, but how people saw. At the MBL, Shinya Inoue, a pioneer in recording live cells, designed his own microscopes and centrifuges. He used a technique with polarized light to better highlight and record cell parts and division. Video microscopy captured the dynamic motion of cells and their parts. Moving images of cell parts changed how scientists thought about cells. It changed how they thought about what cells were doing. Instead of static and rigid, people began to think of cell parts as dynamic and active. To study cell parts, scientists chose to focus on single cells. Research that emphasizes the parts of single cells or the behavior of a single cell reflects the choice to treat one cell as an individual entity. But out in the world, cells are rarely found as individuals. It is becoming more popular in microbiological research to treat cells as members of aggregates or communities, whose features and behaviors are more important than those of single cells. At MBL, researchers Scott Chmielewski visualize aggregates of cells of the mouth, learning about spatial relationships. Does this emphasis on aggregates suddenly make all the research on individual cells wrong or inaccurate? Of course not. Just like thinking about cells as an individual entity is a choice, so is thinking about cells as members of groups. Throughout the history of cell biology and microbiology, researchers have thought about cells as individuals, as members of groups, and even as both at different times. Scientists' ideas about cells were shaped by their specific questions, technologies, and prior ideas. It is not more accurate to think of cells as individual entities, nor is it more accurate to think of them as aggregates. Rather, scientists might learn different things by thinking about cells as aggregates or as individual entities. Today, scientists want to design their own cells, like cells that are made of man-made material and organic material, or cells that have the minimal number of genes to survive. What's important to know is that scientists are making choices. At the MBL, faculty researchers Dr. Carrie Alberton and Josh Rosenthal teamed up with summer researcher Professor Karen Crawford to make amazing discoveries by disrupting the genes of cephalopod embryos using a technology called CRISPR. In their first study, they modified a gene that is important in creating the pigment found in squid skin and eyes. By injecting these CRISPR reagents in the first cell of an embryo, they can create squid that lack pigmentation. Going forward, they are using this approach to study the role of other cephalopod genes in the nervous system in development. Remember, we learned that thinking about cells as aggregates is not necessarily more accurate, rather, it's a choice. Similarly, the features of a design cell are a matter of choice. How do scientists choose what features might constitute a successful cell? Their ideas about what a cell could be or should be are influenced by their current ideas about cells. If scientists in the year 1888 tried to design their own cells, those cells would be very different from the cells scientists want to design in 2024, because their ideas about what cells are were very different. What is a cell, and how do we know? I hope I have helped to improve your understanding and inspired your imagination. You learned that the answer to what is a cell depends on who you ask and when. You learned how do we know is a combination of seeing, technology, ideas, and images that are continually changing in tandem with one another. Researchers at the MBL inherited their ways of seeing, their technologies, and their ideas about cells from past scientists. Many researchers today are exploring new ways to imagine and design cells through synthetic cell biology. The next generation of scientists will inherit their ideas about cells from today's scientists. What technologies, ideas, and images do you think will be part of the next generation?